There you go. Thanks, Guy. Thank you, Dr. Timlin. I appreciate that, and I appreciate being able to address some of the topics in here. And what's interesting is some of the questions that's been asked. Hopefully, we address some of them as we go through this. And what I'm hoping to do in this presentation is kind of talk a little bit about what value we get from things, but then also share, building off of what Tom talked about in the last presentation, on some of the future research that's being done to hopefully add more value to a breeding soundness exam. And so the first question we really have to ask ourselves is, why do we test bulls? We've talked about developing bulls if they're developed properly. We've talked about feeding them in the off season and managing them. Why do we really need to test bulls? Well, the big reason is reproduction is 5 times more important than economically than growth and performance, and 10 times more important than most product quality. And so when we think about what they provide to your operation, it's important to have as good a fertility as possible. And so then we really get into asking, what does that bull do for us? And he provides the genetics to the majority of the calf crop. And so if we do the simple math on it, if a bull sires 30 to 60 cows or calves per year, if he stays in the herd 4 years, he's providing half the genetics to somewhere between 120 and 240 calves. And so with that being said, he's having a huge impact in not only this year, but for several years to come on your operation. And so we want our best bulls to produce the most offspring they can. We also need to realize if we lose fertility, and that question was asked to Dr. Gary, is how many bulls fall out each year? Well, we know that if they fall out and they're not tested, then we end up losing a calf crop or losing part of a calf crop, and that has huge economical benefit or detriments to our operation. And some more research that was done shows that about 20% of unselected bulls are actually subfertile or infertile. And so when we look at those things, we know there's a huge economical benefit to removing low fertility from our herd. So how's that test? We've heard it referenced in several. Dr. Gary put up a few things I'm going to expand on a little bit on looking at a breeding soundness exam. What is a breeding soundness exam? And I don't want to take everything. If you continue on into the presentations later on today, we're going to have a total discussion specifically on the breeding soundness exam and the details in it on the veterinary side. But there's 3 main parts to it. There's the physical soundness, evaluation of semen, and the evaluation of production, or the scrotal circumference, because scrotal circumference is really an indirect measure of how much sperm that bull can produce each day. The physical exam is really looking at the bull. Now, I've got a slide here in a little bit that talks about how many bulls fail a breeding soundness exam for different reasons. And the studies that have looked at it show a breeding soundness exam, most bulls do not fail for a physical exam. Because when we look at their eyes, their structure, things like that, most producers are going to recognize those, know they have injuries or things like that, and they're never going to make it to a breeding soundness exam, or they shouldn't. And so a lot of bulls in this criteria never even get tested. So the more important parts to it, if you're watching for that, is what's next. Well, everybody knows scrotal circumference. It's something that we've made genetic selections for years. We've seen tremendous growth in it. We've seen benefits of it. So a lot of those benefits really come into what impact does it have on their offspring. We know it's highly heritable, and so bulls with larger scrotums have sons with larger scrotums. They also produce daughters that reach puberty at a younger age. And so it's one of those traits, even well before genetics were known, that we actually can go through and select for and have selected for and have tremendous benefits on the fertility of our herd. Now, it was mentioned earlier, the minimums that are required. And we know there's differences in breeds and differences in their ability to, or growth rates on scrotal circumference. But the Society for Therapeutic has set minimum guidelines based on age. And Tom did a really good job of showing the growth that even they saw over short periods of time for scrotal circumference. And so those guidelines are set as minimum requirements, and they're on the low side. And so we need to select bulls that meet these requirements at least. But the area that I really want to focus more on is on semen quality. Within semen quality, there's a lot more affected on the progressive and morphology of sperm. And we see the 4 different measures that should be evaluated. And when we look at those minimums, Tom talked about bulls that just barely pass them. Are they doing us any good? Well, it really depends on the bull, and it really depends on the situation they're in, because we know that all of these things can change over time. We know we've had the talks already talking about nutrition, how nutrition can impact semen quality, but also temperature, injury, any disease can have impacts if we're short term on semen quality. And so it's important that we know where the bull is at and what situation he's in to be able to go through and evaluate what the situation is. So if we look at motility, here's a video from actually the CASA, and Tom had talked a little bit about what a CASA will do. And we can see the differences between motile sperm and progressively motile sperm. Those sperm that have a blue highlight on their path are considered progressive sperm. So they're following a forward momentum in their movement. Those are the sperm that are involved with fertilization. And so that's the reason in a breeding soundness exam, it's important to be able to distinguish on progressive motility. We see several other sperm on the slide that are moving, but they're not moving in a direction that will allow them to be involved with fertilization. The other part that really comes into play is morphology. And Tom mentioned this, that passing a bull that meets the minimums that may be subfertile, thinking he's going to get better, is not doing us any good. Well, one of the things, this is a study done several years ago where they actually took bulls, and the random group is bulls that passed the breeding soundness exam, or the other group that they compared them to is they bumped morphology up to 80% normal sperm or greater. So that's a 10% increase in the requirement for normal morphology. But what we can see over 2 years with quite a few animals in the groups, a 5 to 6% increase in pregnancy rates in those groups that had higher morphology. And we know from work that has been done in the past that morphology plays a tremendous role in early embryo development, in fertilization ability, and in getting animals to breed. And so the more selection pressure we can put on bulls, the better off we are in being able to put as much pressure as we can on fertility, trying to select out bulls that are going to be more important or have better fertility. Now, I mentioned that I was going to put up a slide on why bulls fail a breeding soundness exam. And this was a study done a few years back where they evaluated almost 4,000 bulls between 10 and 19 months of age. There was 10 different breeds and 5 different test stations. And what they really came up with was only about 10% of bulls failed without the semen collection. So 7% on scrotal circumference, 3% on physical abnormalities. Like I said earlier, those physical abnormalities, a lot of times, they're called out before they ever reach this point. Scrotal circumference, most people can look and visually appraise the scrotal circumference. And so then we see a higher percentage, 14%, were classified unsatisfactory because of motility, morphology, or both. But in reality here, so there was about 24% of bulls that failed the breeding soundness exam. But as Tom mentioned in the previous presentation, just because a bull passes a breeding soundness exam, does that mean he has good fertility? And the answer is no. And so I had the opportunity, I worked with Tom up at Fort Keogh for 2 years. And this was a set of bulls that was actually part of the study when I was up there. And they were all bulls raised on the station, collected on the station. We looked at all of them under a microscope. They had good motility, good morphology. And when they were put into practice, especially in a timed AI program, we can see these differences. We see a bull that ranges from less than 20% conception rates to bulls that are up 60 or greater. And so everybody wonders, well, if you have this data and can look at it, why can't you tell which bulls, or what was the difference between them? Well, there's lots of things that come into play, and Tom set this up very nicely with all the work they've been doing, looking at the different measures they can over the flow cytometer. Well, one of the questions that was asked was the difference. Most of the time we do things on frozen sperm, and we know that freezing sperm changes that sperm. We know it kills a large percentage of sperm, and so what we have left are sperm that will survive the freezing process. So we were interested in looking at this a few years ago, and we worked with Select Sires, and what they did was they collected bulls, and there's 6 different bulls in this study. We used them either as fresh semen that was packaged as 2 million sperm per straw, 10 million sperm per straw, or the frozen sperm, which is normally 20 million packaged, so you end up with about 10 million live post-thawing. If we look, there's tremendous differences in pregnancy rates between the 3 treatments. If you look, bull 1 showed no real difference between treatments, but bulls 2, 4, and even 5 and 6, the frozen semen actually had better fertility than the fresh semen. Well, we wanted to go back and look at this and evaluate some of it on the CASA analysis. And we can see when we look across it, motility, progressive motility, velocity, straightness, morphology, we see no big differences between these bulls. And so we have to start thinking about what we're measuring as we go through these things, and the differences that we can pick up, and what we know and what we don't. And so we know motility is extremely important, and every one of these bulls would pass the minimum requirements. There are differences, bulls on the lower side or higher side, but they all meet minimum requirements, and we don't see anything that percent progressive or percent modal, once they meet those minimums, is having the measurable effect. And so one of the things we really need to remember on a breeding soundness exam when we do this is it's really a snapshot of a bull that day. When we collect that semen sample from him, it is the semen that would be ejaculated the day it's collected. And we have to realize that spermatogenesis in the male is a continuous process. And so when we look at the spermatogenic cycle in bulls, it's a 61-day cycle. And one of the things I think a lot of times we forget when we're talking about fertility in bulls and we're talking about what we're measuring is if we're measuring the sperm that are used during fertility. Because we know, I mentioned earlier, things can change over time. Nutrition, disease, injury, all of these things impact spermatogenesis. The discussion earlier, bulls that get fat change semen quality because they can't dissipate the heat as well. And so we know it's important to consider things as they are being used during the breeding season. There's also some traits, if we're looking at natural service, that are not being measured in a breeding soundness exam, because we do not have a good straightforward way to measure these. One of them is mating ability, and that's the ability to actually be able to service a cow. And so several of these things are pulled out in the physical exam, but looking to see, can that bull actually mount and breed successfully? The other thing is serving capacity. We have different guidelines for recommendations for serving capacity. Typically younger bulls, we recommend lower rates. Older bulls would be at greater rates. But in reality, we don't have a good measure. I know of one study that was done at US Mark that actually, where they did parentage testing, and a young bull actually outbred all of the older bulls that he was compared to. And so we don't have good measures of that. We really just try to adapt them to the animal and what the semen concentrations are. We also know we can synchronize with natural service, but that puts greater pressure on the bulls, and especially bulls that have a lower serving capacity. They're going to have greater pressure. Where can they handle the increased services in a short period of time? We also wonder, when we turn multiple bulls out into a breeding pasture, how do we measure social dominance? There's several studies that have been published that shows the dominant bull is going to breed the majority of the animals regardless of the numbers that are out there. And that your less dominant bull is going to breed very few. Well, we always need to be cautious if something happens to our dominant bull, and it's not always the oldest or the biggest bull that is the dominant bull. And that leads us into the other thing, libido. Libido is actually the willingness of a bull to mate, and it's highly heritable. But how can we actually measure this? The practical way is for you to pay attention when you turn bulls out. So it's practically measured by watching the bull after he's introduced to the herd, and what is his main interest when he's turned out with the cows. I was fortunate a few years ago, I was on a meeting with Dr. Barth from Canada, and he shared the next couple of pictures with me. And he said the main reason he kept them was because most people didn't believe that it would occur this way. But they actually did a lot of work measuring libido. And if you look, these two bulls are turned out at the same time. This cow is stanchion, she's in standing estrus, to observe the interest of the bulls. And I usually ask people, when I can see their reaction when we do meetings in person, what do you think is going to happen? Both of these bulls are approaching this cow, and which one do you think is going to be the most interested? When we look at this, even though the bull on the left, much bigger, appears to be older, he's by far not interested. And Dr. Barth made the comment that they really kept this bull around as a demonstration to show whether bulls always had an interest. He said his semen quality was just fine, but in a natural service situation, he had no interest in breeding cows. And so when we think about the breeding soundness exam and the value in it, its biggest value is it's a tool to detect sterile or infertile animals. We still need to evaluate things as we turn bulls out for natural service. A bull that is below average or even low fertility can get classified as satisfactory. And that's what Tom mentioned. The bull that passes because, oh, it's early in the season, we think he'll get better, he's young, things like that, is not really doing us any favors. We need to put as much pressure as we can. We need to know that just because a bull passes a breeding soundness exam, high fertility is not guaranteed. And so what are these other tests? What can we do? And Tom talked a lot about them on that we can measure to further explain bull fertility. And the fact is that it's not going to be a single trait or a single thing we can measure, but are there multiple things? And there's been several tests that are developed that really don't tell us more than what we ought to already know from a breeding soundness exam. And hopefully we'll get to the point that we learn traits that do add to what we learn from a breeding soundness exam. Now, Tom introduced this study. Salo Zoka, who did his master's with Joe Dalton, the University of Idaho, is working on a PhD with me. And this paper and this study was part of his master's program. And what they did is they had these 5 different bulls, and they bred them to almost 5,000 Nelore cattle in Brazil. And as Tom pointed out, Bull A and B had the highest fertility, and bull C had the lowest. Now, one of the things they did after they measured that was actually looking at all of the traits measured on a CASA. And so, as Tom mentioned, there's several different traits measured on a CASA that have to do with motility. And if we look down these, A, B, and C, who are the two highest and the lowest, really don't stand out. So the green are the bulls that were the highest in that trait, and the red are the bulls that were in the lowest. And so bull C, who actually had the poorest pregnancy rates, actually did not stand out as the lowest in anything. And so this is an important thing for us to realize is when we're looking at bulls used in AI programs, they've already gone through selection criteria. They've already met minimum standards. And a lot of these things don't add to what we know. And so what are some of those differences? Well, one of the things, and the question was asked to Tom, what about the changes in the uterus? What about changes in the environment and how bulls can handle that? Well, we were really interested in one of the things we agree that we think is having a big impact on fertility of bulls is actually longevity of semen. And so we worked with select sires on the study, and we purposely went out and we bred cows everywhere from 30 hours before the onset of estrus to 30 hours after the onset of estrus. And if we look at this, bull 1, if we could all select bulls like him, timed AI would work great because he had similar conception rates everywhere that we used him. Now, what's interesting is bull 2, if we bred him prior to the onset of estrus, his conception rates were extremely low in that 20 to 30% range. Nobody's happy with that. But once we followed the AM/PM rule and we got to 18 hours after the onset of estrus, look, he jumped up to over 70% conception rates. And so we know that longevity, or the ability of the sperm to live in the environment it's put in, has a huge impact on fertility. And if we look down below, we did measure several things. We measured membrane viability, DNA stability, motility on the CASA, again, both total and progressive motility, either at thaw or 3 hours later. And we can see there's really no difference between these bulls in any of those measures. So we have to think about sperm transport, and we have to think about what is involved in fertilization as we go through and measure some of these things. And so when we think about what we hopefully the future will hold, is we know things are important. We know progressive motility, normal morphology, intact membranes, the stability of the DNA, ability to undergo capacitation. We have tests for these. We know they are very important in fertilization. But we also know they don't account for all the variables that explain differences in fertility. And so when we start thinking about what we ask the sperm to do, there's other things that come into play. And two of them I want to talk about are surface proteins and microRNAs. And when we looked at the longevity of semen, and we know there's differences, we wanted to begin to look at what can be affecting longevity of semen. And so the best place to look at that is we know semen is stored in the epididymis for extended periods of time. Well, with it being stored in the epididymis and still remaining viable and able to fertilize, we wanted to compare that to what happens after ejaculation and what changes can be occurring. And so we did a study where we actually collected bulls, and then we also collected semen post-slaughter from their epididymis. And this graph kind of shows the differences. There's quite a few differences. This is in the fluid, so the environment in which the sperm is being held in. In the epididymis, there was 208 proteins that we were able to find that did not occur in the ejaculate, and we can see there was 75 added in the ejaculate that did not occur in the epididymis. If you look at the take-home message, a lot of those had to do with mitochondrial function and motility. And to kind of show how that occurs is Tom mentioned the mitochondrial membrane potential and how much energy the sperm can produce. And if we take both ejaculated and epididymal sperm and we put them in the same environment, the same culture media, we can see that there's increased fluorescence intensity, so increased membrane potential or increased ability to produce energy from the epididymal sperm. And if we take it down to the level that it's stored in the epididymis at a pH of 5.8, we can see it is stored even more. So there's even more potential there. And so we know the environment that we place sperm in has a tremendous impact on its ability to fertilize and how long it will live. The other thing we were interested in is what changed with the sperm. And could these changes actually impact sperm transport? And so, very similar graph to the previous one, the difference between epididymal and ejaculated sperm, but these were proteins that were bound to the sperm. And we can see there's huge differences between them too. But one of the big things is on sperm, a lot of the proteins are involved are proteasomes, or they're used to degrade proteins. Well, you may question, why are proteins on the sperm involved with degrading proteins? Well, when we look at a few of these, and these were proteins that were found only on ejaculated sperm, two of them really stand out, and I've got them bolded there because they say the cleavage occurs in an acidic pH. Well, when we talk about an acidic pH, that's important because we get changes in the uterine environment around the time of estrus. And one of the things that needs to be cleaved is you have to realize that sperm has to penetrate the cervical mucus through the cervix to be able to get to the site of fertilization. Now, granted, with artificial insemination, we bypass the cervix and we deposit the semen into the uterine environment directly. But we also get changes in the uterine environment. And this is some work we did several years ago where we actually measured the uterine pH at different times. And time 0 there, which is the lowest point for both the green and the yellow, is the onset of estrus. And we compare that to cows that did not exhibit estrus around the time of AI, and we see their uterine pH did not change. And so, depending on the environment we put that sperm into, we can have huge impacts on its ability for duration of survival and also changes that will occur in it. And so, with knowing that the proteins on the sperm surface are important for transport, one of the things Saulo is really interested in his PhD is, can some of these proteins be used as a marker for fertility? And so some of them he's started with is both CD9 and serapina A5, which are just two proteins. They're localized on the sperm head. We know they're involved in cell-to-cell interactions and binding. And so his interest is, can these be involved with the sperm reservoir? Now, one of the things originally going in is the amount of protein or the absence of it on the sperm head important? But as he started looking at things, it may not just be intensity. And so you could tell in the previous slide in those pictures that sperm fluoresce greater if they have more protein on it. But one of the things he's noticed was that, like, serapina A5 was also detected along the sperm of the tail. And so about 33% of the sperm tails were also fluorescing. And what was different is that range between bulls. And we can see in this graph somewhere between about 4 and 60% of the sperm tails between different bulls had fluorescence attached to them. And so when we think about that, it may not just be how much of a protein is there. It could also very well be the location of the protein and what impact it can have during sperm transport. And so, as Tom mentioned, one of the hard things to do with this is trying to get bulls that we have known fertility data from the field with. And so another student has looked at, well, if we're looking at sperm transport and we know there's differences in sperm transport, what impact does that semen have on early embryo development? And so we were able to get semen from select sires that had known good fertility or lower fertility. All of the bulls used in this, there's 23 bulls. They had they averaged 5,000 inseminations per bull over multiple ejaculates. And so with that, we're able to look at field fertility data coming back from AI protocols. And what Steph was interested in was looking at the microRNAs in the sperm. And when we're looking at this, a lot of times we're thought that, as I mentioned earlier, the sperm's providing half the DNA to the future calf crop. And so as we're looking at what's going on, we think about growth traits and carcass traits and all of those things. But the DNA for that is not the only thing the sperm takes into the egg when it fertilizes it. These microRNAs are actually small messages that really don't code for any gene expression, but what they can do is they can bind and change the expression that's there. And so they'll end up turning on or turning off different genes as that embryo is developing. Now, as I mentioned, we looked at 23 different bulls classified as either high or low fertility bulls. And what we saw when we grouped them based off of sire conception rates, there was 10 microRNAs that were different between these two groups of bulls. There was 506 that were similar, but only 10 that differed. And when we look at these, we see that it didn't matter. We see some both up and down regulated in our low fertility bulls. But what we do know is some of these microRNAs have been shown in other species to actually impact embryo development. And so if the family of microRNAs is missing, embryo development is actually stopped. And so when we think about fertility in a bull, it's not just sperm swimming to the site of fertilization. We have to get it to the site of fertilization, but then we also have its impact on embryo development. And embryonic mortality is one of the things that then can have negative effects on that sire's conception rates, because a lot of times these embryos die even prior to us being able to determine early pregnancies. Well, Sofia Ortega at the University of Missouri recently published a paper where she took bulls with sire conception rates and did IVF to look at embryo development, to actually look at what impacts the sire or the semen would have on early embryo development. And we can see here 2 sires that were classified as high sire conception rates, 3 sires as low sire conception rates. We can look at cleavage by day 3 or blastocyst by day 8. And we can see when we look at cleavage by day 3, there's really no difference between any of the bulls. But as we go out to blastocyst by day 8, we see the high fertility bulls had a higher blastocyst rate than the low fertility bulls, but there's actually one of the low fertility bulls that had just as good a blastocyst rate by day 8 as the high bulls. Well, we actually looked at these 5 different bulls with the microRNAs, and one of the microRNAs we can see nice lineup as our high fertility bulls are positive, our low fertility bulls are negative, but they don't all relate that way. We see others where it's kind of all over the place. We also see some where the one bull that had good embryo development to day 8, but was classified as a low fertility bull, is the opposite of everybody else. Well, Sophia in her paper actually investigated further out all the way to hatching what impacts we're able to have. And if we look at bull 8, who had good development to day 8 or blastocyst formation had the poorest number of cells or the lowest number of cells in the trophectoderm, or what becomes the placenta, by the time it hatched. And so we have to realize, even when we're looking at embryo development, there's several things that come into play, and it's not just can they develop quickly, because that one fertilization, that single cell then differentiates into the placenta, into the embryo, and all the parts that go into it. And so this becomes a very complex situation, and we need to look at all of these different things, and it's being able to have the data not only from the field, but how that embryo develops and sperm transport to be able to build this information that Tom was talking about and what impacts they may have on a bull. And so we know there's differences between high and low fertility bulls in these microRNAs. They support the idea that these microRNAs can have a direct impact on embryo development. And so as we think about it in those situations, there's several things we can measure. Now, to go back to the original question I was asked as the presentation, was the value of a breeding soundness exam. The breeding soundness exam, I think, is always going to be the first step in bull fertility. All of these other measures that Tom talked about with a flow cytometer, that I've talked about with proteins, with microRNAs, are a much more in-depth analysis. A breeding soundness exam, it will allow us to always remove sterile or infertile bulls. It allows us to make that first selection pressure to have as good a fertility as possible. The other thing is, what's done in a breeding soundness exam can easily be done chute side, can easily be done in the field where there's not really electricity, not anything major. A lot of these other tests require a lab, require molecular techniques. And so it's one of those things that as we go further and we want to identify the most fertile bulls or remove off more subfertile bulls, they're going to be much more in-depth tests. And so when we think about a breeding soundness exam, they are always going to be that first step in removing things. Just like I mentioned earlier, that physical traits is one of the lowest reasons bulls fail a breeding soundness exam, because producers can identify that at home before they ever go get them tested. Our next step is always still going to be that breeding soundness exam, in my opinion. And then hopefully we develop the technology and the markers to look for what can be next to remove those things. With that, there's lots of people that have helped with these, lots of collaborations, from continuing to collaborate with Tom up at Fort Keogh, University of Missouri, USDA, US Mark in Nebraska, and my former institution at South Dakota State, lots of grad students. And then, of course, none of this would be possible without the funding and the industry that has helped with all of these different studies. And so with that, I think we've got time for a few questions, and I'll be happy to answer what I can.
All right. Thank you, George, for that very nice presentation. So reminder, go ahead and put your questions into the Q&A box so that we can get those answered. George, the first question that came in here is, you stated that scrotal size was important for several reasons. Can you just expand a little bit on, is there such thing as too large of a scrotal size, and does that impact sperm production?
So one of the questions do get asked is, can scrotal size get to be too large? And it really depends what that reason is. I have seen scrotal circumferences very large where a bull has been injured. And so that's the other part of scrotal circumference, isn't just measuring the size, but also palpating the testicles. And sometimes the testicles will swell. And if that's happening and they're soft and mushy, then yes, there's damage that's occurring. And usually if they get to that, then they'll start to degenerate, and then they'll start shrinking in size. And so I haven't seen any data that says just a large scrotum has any negative effects unless it's been injured, which is causing it to be large. And so if you visually see a bull and he has a really large scrotum, The biggest thing there is making sure he hasn't been injured for that reason.
Excellent. Also, so just back to, thinking about Tom's talk and the markers that he talked about, the markers that you mentioned. So has there been any cross-validation of those markers on the similar sets of bulls, let's say to, if you go back to Tom's data looking at their markers on the flow and looking at them in your microRNAs, just trying to find the most optimal, let's say, marker that could be used for bull fertility?
That's one of the things we're actually working on right now. Actually, Salo is actually in Tom's lab right now, working on some of that with those bulls. So to collect data like that takes a lot of collaboration, takes lots of data coming back. And so working with the AI industry and labs working together. And so it was actually very handy that Salo could take the bulls he used from his masters and go to Fort Keogh and run them on Tom's flow. He's run them on our CASA and be able to look at some of those things. And so that work is being worked on. The first thing is having the field data to begin with. And so hopefully over the next couple of years, we do generate a lot more data where we're doing some of that. We're also working with Sophia on doing some more embryo cultures and trying to get all of those things put together to evaluate bulls.
So a question that just came in, any docility score correlation with libido, high docility, bull APDs, low libido, question mark?
I am not aware of anybody who has shown direct correlations between docility and libido. We know docility does have an impact on reproduction, stress does, but to say a bull that has greater temperament or is calmer, has more libido, I'm not aware of any data that has looked at that. It's really the best data that's out there, both in bulls and rams, would show bulls that have high libido, a lot of times their sons will. So they do pass things like that along.
Okay, last question here is, what do you think about incorporating some type of ultrasound measurement into a BSE?
Actually, we just did BSEs here on the station on Tuesday, and we were even talking about that because in addition to palpating the testicles, sometimes when you see things, you can actually see calcification or problems in the testicles. There's studies that have also shown the thermal imaging that was shown earlier to impact some injuries in that. And so I think anything like that is adding data to what we know. And a lot of this, as Tom mentioned, there's not one single trait that's going to be the magical one to say this bull is more fertile. And so it can help you diagnose injuries or things like that, but does it add anything to have a bull be more fertile? I'm not aware of any of that, but any information helps you weed out those bottom or subfertile bulls, definitely.